The overall goal of this procedure is to extract high molecular weight DNA from hypersaline microbial mats. This is accomplished by first homogenizing and extracting microbial cells from the microbial mat. The microbial cells are then lysed using freeze-thaw and chemical methodology. Next is the removal of cell debris and RNA using organic extractions and RNase, followed by DNA extraction. The final step of the procedure is DNA precipitation and purification. Ultimately, results can be obtained that show DNA of high molecular weight, quality, and concentration.
The main advantage of this technique over existing methods such as direct and harsh DNA extraction methods is that it yields high molecular weight DNA from a complex environmental sample for functional metagenomic studies.
The microbial mat used in this study was obtained from a hypersaline pond located in Eleuthera, Bahamas. To extract microbial cells from the background mat matrix, first mix approximately 30 grams of mat thoroughly, without grinding, with a sterile grinding pestle. The mat is thoroughly homogenized when no lumps can be seen in the mixture. Next,